My name is Alicia Hernandez. Today is Wednesday, October 6, 2021. This is Lab 9 and this is for Bio 225. So prior to starting this experiment, I already washed my hands thoroughly with soap and warm water, and I also wiped my area down with 10% bleach, and I already put on my PPE, and I already labeled, I used a permanent marker and I labeled all these tubes, the broths. And there's two parts of this experiment. Part one is the salt testing, and for this one I'm gonna add 2 drops of the nutrient broths of the staph and the yeast, and then part two I'm gonna add 1 drop of the sodium hydroxide to the pH 9, and then 1 drop of the HCI, sorry I couldn't pronounce that, to my pH 5, and the 7 one stays neutral. So I'm gonna start already. So I'm going to, and I already have my pipettes and the alcohol too, and my candle on. So I'm gonna start with, I'm actually gonna start backwards. I'm gonna start with the nutrient broth 7%. I'm gonna start with my yeast. So I'm gonna open it up. Cap right here, and I'm gonna sterilize the lip. Put it back right here. Gonna grab that yeast nutrient. Gonna take off the cap. Gonna sterilize the lip. I'm gonna grab my pipette. That's in alcohol, and shake it, make sure it's completely dry. So I'm gonna get 2 drops of the nutrient broth of the yeast. Gonna put them in here, 1, 2, and then I'm gonna put this on the bleach, my bleach solution. So I'm gonna sterilize the lip again of the nutrient broth of the yeast, and then put the cap back on. Gonna sterilize this because I put the wrong one. My tube again, sorry. Sorry. Okay, so now I'm gonna grab the yeast and I'm gonna sterilize the lip. And then put the cap back on. Alright, so now I'm going to grab the nutrient 7% of the staph, take off the cap, and sterilize the lip. So I'm gonna repeat the same step, put it back in here, and then grab my staph, nutrient broth, and then sterilize the lip. And then I'm gonna grab my pipette, flush the alcohol, make sure it's completely dry. And I'm going to put 2 drops in here, 1, 2, and put this on the bleach solution. So I'm gonna sterilize the lip of my nutrient broth, and then put the cap back on. And then sterilize the lip again. So I'm going to repeat this same step to my other nutrient broths, to the 15% and to the 1%, the same step. So now part 2 of the experiment, which is the pH testing. So I'm gonna put 1 drop of the sodium hydroxide into the yeast pH 9 and the staph pH 9. So I'm going to open this up and then sterilize the lip. And then I'm going to put just one drop. Okay. Going to sterilize the lip. And put the cap back on. So now I have the staph nutrient broth of the pH 9. Gonna sterilize the lip. And then I'm gonna put one drop again of the sodium hydroxide. Okay. And sterilize the lip again. And put the cap back on. So the pH 7 is gonna stay neutral. And then for the pH 5 of the epi and the staph, I'm gonna do the same thing. I'm gonna put one drop of the hydrochloric acid of the HCl. And so I'm gonna wait 24 to 48 hours for the results. And then I'm going to wipe down my work area with 10% bleach, wash and return other items to my kit for future use, and wash my hands thoroughly. I already said that, okay. And that's it. And then put anything that needs to be in the bleach solution, and then we're gonna wait for the results within 24 to 48 hours. And that's it.